Welcome everyone, my name is Salvatore and I'm a postdoc of the Department of Systematic Botany at the University of Göttingen in Germany. I will be talking today also about extraction methods on herbarium specimens, but from a bit different perspective. I will tell you something about ancient DNA extraction methods applied to old herbarium specimens. I will give you some information about costs and advantages of using these methods, and finally give some consideration on when it's worth to invest more time and money for using those methods in herbarium specimens. Herbarium are a rich collection of plant biodiversity, and for systematists they are potentially a huge source of data. We have in a single place plants from different species collected in different regions of the world in different periods, and for example we can work with extincted species. And last but not least, since we are part of the taxonomics, herbaria houses types, and types are extremely important for study having taxonomic purposes. If you want to work with herbarium specimens, and especially if you want to apply DNA-based techniques on old herbarium vouchers, you have to deal with some issues. First of all, there's not much DNA on them. The few DNA present then is also degraded, so it means it is very fragmented and also partially damaged or single-stranded. There are also chemical modification happening spontaneously on old DNA, like cytosine deaminations. And finally, you have really to be careful with contaminations. So contaminations are always an issue, but since in old museum specimens the DNA is so few and so damaged, the effect that contamination can have on the downstream molecular analysis is really, really bad. In plants, it seems to be even more complicated. So there are studies claiming that DNA fragmentation is 6 times faster in plants compared to animals. So basically, working with a 200 years old herbarium specimen is like working with human remain from early middle age. To choose the right extraction method for your herbarium specimens, you have to consider a few points. First of all, you want to choose the method that is able to extract as much DNA as possible from your specimen, because as I said before, the DNA contained in those old herbarium vouchers is not much. You want also to choose the method that increases as much as possible the proportion of small fragments of DNA extracted. Usually extraction methods tend to extract rather longer and good quality fragments, but well, in old specimen you have only small fragments. And you have to follow a list of rules in order to avoid as much as possible contaminations. So you have to work in a separated and clean lab, possibly under a laminar flow bench. You have to wash everything, surfaces, but also consumable with bleach or irradiating them with light. And then you have also to follow some specific hygiene rules, like wearing overall coat, masks, glasses if possible, and so on. If you give a quick look at the literature, you will see that people have applied mainly two strategies. Either they used standard extraction methods for plant material, like different kits, or the setup protocol, and then with few adaptations, like a prolonged lysis time, or using all those hygiene rules I've just talked about. Or the other strategy, they applied methods that are usually used for damaged DNA to herbarium material, so like method used normally in forensics, or method used for archaeological remains and environmental DNA. An example is this study by Gutaker and colleagues. They adapted a protocol described originally by Dabney and colleagues to, the method was described for archaeological remains, and they adapted it for plant materials. and then they compared it to the standard setup protocol. To test the efficiency of those two methods, then they used Arabidopsis thaliana, and they used several herbarium voucher, all of them collected more or less around 1820. Well, the main modification was the addition in the lysis buffer of those two compounds, the PTB and DTT. The PTB cleaves glucose-derived proteins cross-links and basically help releasing the DNA trapped in the sugar-derived compounds, whereas the DTT, the dithioteritol, reduce disulfide bonds and help releasing the thiolated DNA trapped in cross-linked complexes. Both compounds have been already successfully used in methods extracting DNA from archaeological remains. We can reduce the whole protocol in three main parts. First, we have a long incubation in the PTB-DTT lysis buffer, so usually it is an overnight incubation. Then samples are filtrated through the Kia shredder columns. So those are columns found in many commercial kits of Kia again. And finally, we have a DNA precipitation using the mini-elute column. This is also a peculiarity because mini-elute columns usually are employed for purification of PCR product and not for extraction for DNA isolation. In this way, the methods can ensure an higher DNA yield thanks to the long incubation in this specific lysis buffer, and also a higher proportion of small and endogenous DNA fragment because of the employment of the mini-elute column. In fact, those columns are able to retain DNA from very small fragment of DNA starting from 50 or 70 base pairs long DNA. If you are interested in the method, or if you want to apply it to your old herbarium specimens, you can take a look to the Museomics on cloud folder, and there you will find a video, an hands-on video demonstrating in the lab step by step all the protocol. Finally, Göttingen and colleagues found out that the PTB-DTT protocol is outperforming the setup method when working with very old herbarium specimens, especially if we consider the proportion of small fragment extracted, and also then when looking at the results of the sequencing, and especially when they mapped the reads to the reference genome, they could see that the libraries produced from the PTB-DTT extracted had a higher proportion of endogenous reads, and that also those libraries were more heterogeneous with a higher number of unique molecules. However, the method has also some disadvantages. First of all, it's more expensive than the other methods, so it is at least twice more expensive than an extraction with the Göttingen plant mini kit, and way more expensive than a setup extraction. It is also more laborious and time consuming. An extraction is lasting 2 days, and you have to consider that you cannot extract so many samples at once like you will do with an extraction kit. Moreover, it's also producing much more waste, and especially hazardous waste, which needs to be then treated in a special way. Well, since I'm not a specialist of aDNA, I'm rather a botanist who is interested in extracting DNA from my herbarium specimen in the easiest and fastest way as possible. So the question is, well, when it is worth the effort to apply those methods. So when it is worth to invest time and the resources to apply those methods in for an herbarium specimen. I tried to answer this question in the first part of my project. I'm working with Xanthium. And my project is actually based on herbarium vouchers, most of which are relatively old herbarium specimens. I did this try first with together with my colleague Natasha Wagner. She has also a project in the priority program, but she's working with a completely different plant genus. She's working with Salix. So we used a herbarium specimen from the two genera, and we included also different species of the two genera. We included herbarium specimen from very different ages, so spanning from vouchers a bit older than 200 years until very recent herbarium specimens. And we extracted DNA from those specimens with the two extraction methods. So one, the Kiagen DNase plant mini kit. This is the standard method used in our lab, and with the PTB-DTT extraction methods. Finally, we measured the concentration as a measure of DNA yield, and the purity of the extract, checking for the absorbance ratios with the nanodrop. And finally, we wanted to compare the efficiency of the two methods, so the PTB-DTT method against the standard Kiagen extraction kit. And then we wanted also to see if there were some differences between those two genera, or between different species within the two genera. In general, we found a better performance of the PTB-DTT extraction methods compared to the Kiagen kit in terms of DNA yield. We didn't find, so when we consider the two genera separately, we didn't find any difference among taxa of those genera, among species of those genera. And we did find a correlation between age and DNA yield. So basically, as one would expect, older samples, irrespectively of the methods, performed worse than newer herbarium specimens. When we compared the two genera, we did find a difference. In both genera, Salix and Xanthium, the PTB-DTT method performed better than the Kiagen kit. However, in Xanthium, both methods produced better results compared to the extractions in Salix. And when we also look at the negative effect of the herbarium specimen's age on the performance of the extraction, we see that this effect is much more pronounced in Salix. For Xanthium, for example, we have samples that are almost 200 years old that produce good results with the Kiagen extraction kit, so almost 20 nanogram per microliter, whereas in Salix it did not make any sense to extract a herbarium specimen predating the 20th century with the Kiagen kit, because they didn't produce any extract at all. When we consider the DNA quality, or the quality of the extracts, of the purity of the extracts, we can see that both methods produce comparable results, or even it seems that the PTB-DTT extraction methods produce even better results than the Kiagen kit. And this is probably due to the fact that this method, so the DNA precipitation in this method is also done through a silica column like in the kits, and this is producing those extracts with let's say high level of purity. Since in our project we are mostly using NGS sequencing, we decided to select a subset of samples, 12 in total, 6 from Xanthium and 6 from Salix, extracted with both methods, and to sequence them using very standard library preparation kits and sequencing kits. Well, we have done so for two main purposes. First of all, we wanted to see if those extracts and the old DNA contained in them would have worked also when using a very standard commercial kit for library preparation. And second, because this would have given us a way to estimate the proportion of endogenous DNA in the extract. So basically, we could then map the reads to a reference genome and estimate the proportion of endogenous reads. For Xanthium, we went even farther. We did also some target enrichment, again using very commercial kits. We used my Bates Coles composite kit, which is basically a Bates kit designed to perform phylogenetic study in the Asteraceae, in members of the Asteraceae family. So it is not a kit designed for Xanthium, but rather for the whole family. And most important, it is also not a kit designed to work with aDNA. So we did target enrichment in Xanthium for different reasons. First of all, because it is the methods I want to use in my project. But most important, we wanted to see if a standard kit would work properly also when using aDNA, so with very short DNA fragment. And moreover, there is no Xanthium genome available yet. So doing target enrichment would have given me the possibility to map the reads to the targeted regions and then to get an estimate of the proportion of endogenous reads. Well, finally, we pulled everything together, all the libraries together, and we sequenced them with an Illumina MiSeq sequencing machine. Those are the assembly and mapping results of the sequencing. I don't want to go very much into detail. I wanted to show you just some significant number. First of all, the high percent of low quality reads in Salix, and this is something we could expect, especially for the oldest herbarium specimen. This is not so in Xanthium. The number are much lower, but this is probably due to the fact that Xanthium samples went through the hybrid capture procedure, so probably the smaller damaged fragment get filtered by the target enrichment procedure. On the other hand, in Xanthium, the percent of duplicates reads is very high, and this is actually something very well known from other studies applying target enrichment to all the herbarium specimens. Well, more important for us was to look at the percent of mapped reads, and we were surprised that the numbers were so high. So in Salix, basically 90 to almost 100% of the reads mapped either to the genome, to the reference genome, or to the plastome. In Xanthium, numbers are of course a bit lower because those are the number of reads mapping to the targeted regions, but still 55 to 70% of the reads map to the targeted regions, and this is actually quite good. And I can tell you that those numbers are comparable to those obtained when applying the same techniques to fresh or silica gel material. I will finish this presentation with a phylogenetic tree. How otherwise? Well, actually the tree is not very important. It is just a fast and dirty analysis. It's a fast tree done using about 800 of the more than 1000 loci of the Bates kit. And I did it just to see if the herbarium vouchers I was talking about today clustered according to taxonomy, and they were not placed randomly in the phylogenetic tree. As you can see, this is the case. The old herbarium specimens here are marked in bold, and they actually clustered according to taxonomy. So here the three Xanthium orientale specimens, and here together with the other, so the three Xanthium spinosus samples together with the other Xanthium spinosum. I included also basically twice the same samples extracted with both methods, and you can see they clustered together, so that's fine. Well, that's normal, but you never know when using 200 years old samples. Well, trying to sum up the results of these small studies, we can say that there is a correlation between DNA yield and age of the herbarium specimens. Of course, all the specimens perform worse than new specimens. The PTB-DTT methods produce better DNA yield and quality in general, so for all DNA, for all the herbarium specimens, but also for new herbarium specimens. But one has also to say that this method is more expensive and time-consuming than standard methods. When we compare the two taxa or the two genera that we have used in the study, we can see that, well, there is a difference. And in our case, so in Salix, the negative effect of the age was much more pronounced than in Xanthium. Coming back to the question of the beginning of the talk, so when it is worth the effort, when it is worth the effort of using those methods specific for ancient DNA with older biomaterial. So unfortunately, I don't have a universal answer. As a rule of thumb, I use the PTB-DTT method for all specimens predating the 20th century, or with this specimen for which I don't have much material and for which I don't have a second extraction chance. But this works for Xanthium and might not work for other taxonomic group. For instance, we have seen that in Salix one should be more cautious and maybe use those methods also with more recent herbarium specimens. So there is an effect of the taxonomic group. Of course, there is an effect of the age of the herbarium specimen, but the age is not the only factor influencing the quality of the DNA contained in herbarium specimen. For sure, there are other factors usually poorly known. For example, the conservation condition, how those specimens were conserved in the herbarium, sampling environment, I don't know, Mediterranean environment or wet tropical forest, that might be very different. And most important, preparation conditions. So how fast was the specimen prepared, how fast was the plant desiccated, were used during desiccation other substances. So all those things are very strongly influencing the quality of the DNA contained in the herbarium specimen, but usually are poorly known. With these nice herbarium specimens, I would like to finish the talk. Thank you very much for the attention. And I will take the chance also to remind you that in the Museomics on Cloud folder, you find a protocol, a file with a protocol of the PTB-DTT extraction method. This is the protocol used in our lab, but there you find cited the original papers with where you can find even further information. And again, in the folder, there is also an end-on video demonstrating in the lab, step by step, the PTB-DTT extraction protocol. So, cheers!